Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hi everyone. First of all, let me introduce myself. My name is Justicia Dellarohita. I usually go by Shinta. I'm a first year chemical engineering student at UPN Veteran Yogyakarta. And today's video is gonna be a journal club presentation video. This is for fulfilling my assignment in organic chemistry class. So here's my journal club presentation. In this journal club presentation video, we'll be discussing about article introduction, article summary, importance and relevance, methodology, results and findings, also conclusion. So first of all, I'm going to be introducing the article that I'm going to be talking about. The article title is Amino Acid Composition and Protein Quality of Commonly Consumed Cooked Food in Nigeria. The author of this article is Oluwafunke Akinbule, Oluseye Onabanjo, Silifatsani, Mojisola Adekunwa, Abidun Akinbule, Samuel Sosanya, and Isolua Afolabi. This article is from the Journal of Food Composition and Analysis with the number of identification, which is 119 2023 105295. Okay, to the next slide. So first, I'm going to be talking about the summary of this article. This study determined the amino acid AA composition and protein quality of selected Nigerian meals. The AA profile of 19 meals was determined by ion exchange chromatography and quantified by high-performance liquid chromatography mass spectrometry. Protein quality was evaluated using AA scores, interaction, essential amino acid index, predicted protein efficiency ratio, and biological value. This study is conducted in order to tackle the problem of malnutrition in Nigeria effectively as a result of the lack of information of Nigeria's food composition database from the commonly consumed foods there. The protein quality indices showed that most Nigerian meals met the recommended score for all indispensable AS except for leucine and phenylalanine, but had low biological protein quality. Strong positive correlation existed among most AAs and AFANG soup clustered exclusively in indispensable, conditionally indispensable AAs and AA group. These findings are essential for updating the Nigerian food database. It can also serve as a baseline for food-based dietary guideline. Okay, so moving into the next slide, which is the importance and relevance of this study. So malnutrition remains as a major public health problem globally, with the highest prevalence in sub-Saharan Africa. Nigeria, being a country with the largest population in Africa, has a high burden of malnutrition. This is the data from UNICEF 2021. About 37% children are stunted and 7% are wasted. These values are higher than the average for the African region, which is 29.1% and 6.4% for stunting and wasting, respectively. The data from Global Nutrition Report 2021. Also, 23% of Nigerian children are underweight, while 8% and 58% of children and women respectively are anemic. In addition, an estimated 2 million children in Nigeria suffer from severe acute malnutrition or SAM, and only 2 out of every 10 children affected are reached with treatment. This is what UNICEF said in 2021. This is really heartbreaking to hear about the news, and this is really a big problem to be solved, not only in Nigeria but globally. Like many other developing countries, Nigeria has made no progress towards achieving the target for chronic malnutrition. Inadequate dietary intake and disease, which are the immediate causes of malnutrition, are important factors that need to be addressed holistically in order to tackle the problem of malnutrition effectively. However, dietary intake studies, which are necessary to identify dietary inadequacy in individuals and population groups before intervention can be designed, cannot be effectively conducted without the availability of accurate food composition database. Lack of specific nutrient data on Nigerian foods required to tackle the problem of malnutrition has made it difficult to accurately identify nutrient inadequacy in Nigerian population, and hence frustrates efforts to design and implement effective intervention programs to improve the nutritional status of Nigerian population. In the effort to reduce the burden of malnutrition, Nigeria launched its first food composition database in 2017. However, this database has limited cooked foods relative to Nigerian diverse food culture. So the data on this first food composition database in 2017 is really limited. And also the nutrients contained in the database were limited to proximate and micronutrient contents, leaving out other nutrients profiles, especially amino acid profiles, which amino acids are really essentials in the management of chronic malnutrition. So on the database, the first food composition database that they launched in 2017, they leave out the most important thing or nutrient that is have to be on the database, which is amino acid, because the amino acid is really essential to tackle the problem about malnutrition. However, there is still lack of information on the amino acid profile of commonly consumed Nigerian meals, which is a crucial information required to combat malnutrition in Nigerian population. Amino acids are the building blocks of proteins. They provide information on the functionality and nutritional quality of protein. The protein quality of food is a major nutritional issue that reveals the potential capacity of the food protein and its ability to provide the appropriate pattern of dietary indispensable or essential amino acid. Similarly, and the relationship between individual amino acids and their group reveal their ability to predict malnutrition and or chronic disease. Hence, they are strong determinants of protein quality. In addition, amino acid score, essential amino acid index, protein efficiency ratio, and biological value have been reported as good protein quality indicators. Inadequate dietary protein quality has been linked with malnutrition, especially stunting. Studies have reported the potential roles of several amino acids in the development of chronic disease such as diabetes, cancers, liver cirrhosis, and renal failure. A typical Nigerian meal is carbohydrate-based, with cereal or tuber being the major sources of carbohydrates and legumes or nuts being the major sources of proteins. The meals are usually cooked with a combination of ingredients from both plant and animal sources. The Nigerian meals are mostly consumed with soup or cooked in stew. This soup or stew is cooked with minimal animal protein such as feed, meat, crayfish, palm oil or vegetable oil seasoning. The soup may include vegetables, and particularly and or melons such as egusi melon. Okay, so moving on to the next slide, we're going to be talking about the methodology used in this study. This study determined the amino acid or AA composition and protein quality selected from Nigerian meals. The AA profile of 19 meals was determined by ion exchange chromatography and quantified by high performance liquid chromatography mass spectrometry. So yeah, during the study, the author were using chemicals and reactions and standards to help determine the amino acids on the selected cooked meals in Nigeria. They use water's ACCQ fluor reagent kits, and then they use hydrochloric acid at 37%. They also prepare the 6 normal with 0.5% phenol. Also 2 solvents which are aqueous tetrahydrofuran and acetonitrile. Also they provide sodium acetate buffer concentrates and pH 3.8 and pH 4.6. The standard solution that been used include leucine, lysine, isoleucine, phenylalanine, tryptophan, valine, methionine, threonine, histidine, proline, arginine, tyrosine, glycine, cysteine, glutamic acid, alanine, serine, aspartic acid, and diluted in 0.1 normal hydrochloric acid. Analysis of standard reference material or SRM number 2383 baby food composite from National Institute of Standard and Technology of Department of Commerce, Washington DC, USA, was used for the assurance of assays. Certified standard reference materials values were above 96% for all assays. Also, to minimize the contamination risk, all glasswares were soaked in 10% nitric acid solution for 24 hours and rinsed with ultra pure water before use. All assays were done using analytical grade chemicals. And then to collect the sample, which is the selected meals in Nigeria, the cooked food selected for this study are the one that is commonly consumed, composite meals and soups that were not captured in the Nigerian Foods Composition Database and existing literature pulled in terms of food and nutrients. They include boiled rice and fish stew, bread and beans, boiled yam and garden egg sauce, beans and corn, avada rice, which is unpolished rice and stew, akara and agidi, which is cowpea and maize-based dish, afang, which is wild spinach and flatted pumpkin leaf soup, panga, which is palm nut soup, beef stew, chicken stew, fish stew, egusi or melon soup, keberi, peeled cooked bean soup, groundnut soup, ala sepo or okra soup, miyan kuka or dried baobab leaf soup, ogbono or wild African mango seed soup, oha, which is African rosewood, and okasi, which is wild spinach soup. The recipes were collected from 800 randomly selected households and for focus group discussion session in Southwest Nigeria using cross-sectional and convergent parallel mixed research design. The respondents were households who had a minimum of 2 years experience of cooking in the Nigerian meals and soup evaluated in the study. Okay, so moving on to the next slide, we're going to be talking about the sample preparation. They actually have the table, which is a standardized recipe of the commonly consumed Nigerian cooked food. But yeah, for each of the 3 times the cooked food were cooled after cooking and homogenized. Approximately 200 grams of each cooked food sample was separately homogenized with the aid of an electric blender. The moisture content was determined and the homogenized mixture was dried in an oven at 70 degrees Celsius for 12 hours and packed separately in plastic bottles labeled and dated. Moving to the next slide, it is the determination of amino acids profile. So to determine the amino acids that is contained in the selected cooked food that is commonly consumed in Nigeria, they undergone 4 processes, which is the fatting of sample, and then hydrolysis of the fated sample, description of the analytical column, and loading of hydrolyzed sample into the amino acid analyzer. So the first process is the fatting of sample. The food samples were defatted using chloroform or methanol mixture of ratio 2:1. About 4 grams of the sample was put in extraction table and extracted for 15 hours in sucklight extraction apparatus. After undergone the defatting of sample, the sample is going to be being hydrolyzed. So about 0.25 gram of the defatted sample was put into a glass ampoule. 7 centimeter cubic of 6 molar hydrochloric acid prepared from 36% stock solution was added and oxygen expelled by passing nitrogen into the ampoule. This is to avoid possible oxidation of some amino acids during hydrolysis. For example, methionine and cysteine. This was sealed with Bunsen burner flame and put in the oven at 105 degrees Celsius for 22 hours. Cooled and filtered to remove the humins. The filtrate was then evaporated to dryness at 40 degrees Celsius under vacuum in rotary evaporator. tryptophan, which was destroyed by 6 normal hydrochloride acid during hydrolysis, was separately hydrolyzed for each sample, which 4.2 molar sodium hydroxide for 22 hours and neutralized to pH 7 with 6 molar of hydrochloric acid. The residue was dissolved with 5 cm cubic acetate buffer. So after that, the next step is the description of the analytical column. Amino acid profile was determined by ion exchange chromatography, as described by Benitez, 1989, using the model 120 phenyl thiohydantoin or PTH amino acid analyzer applied biosystem from Thermo Fisher Scientific Waltham in the United States. The amino acids analyzer uses AC18 HPLC cartridge column, slurry packed with porous 5 micrometer octyl decylcyl type sorbent specifically selected for PTH analysis, 22 cm long and a diameter of 2.1 mm ID. The next one, the loading of hydrolyzed sample into the amino acid analyzer. About 60 microliter of the hydrolyzed seed was then injected into the amino acid analyzer for separation and characterization. Quantification was obtained using high-performance liquid chromatography, mass spectrometry by comparing the peak area of the amino acids with the external standards. The peak area, which corresponds to the concentration of each amino acid, was calculated from the integrator attached to the analyzer as the following. So the following slide is how the peak area, which corresponds to the concentration of each amino acids, was calculated. They use this equation, which is concentration G per 100 gram protein equals to NH times width at NH per 2 times STD times C. And then where C is equals to dilution factor times 16 per sample weight in gram times the N% times volume loaded, and then it's gonna be divided by NH and then times by W or W. Also, the other equation is the SSD. NH means net height in millimeter, width means width at half height millimeter. SSTD means value of the standard in the concentration of amino acid loaded into the amino acid analyzer. C means concentration of amino acids loaded into the amino acid analyzer, and means fat-free nitrogens, and W meanings about width of norleucin at half height millimeters. And the last one is Ne, which is norleucin equivalence in the standard micro AA or micromole of amino acids in the standard. Okay, next, the amino acid concentration in dry weight was converted to fresh weight using this equation that is mentioned down below. Okay, so now we have in the presentation about amino acid score. The quality of dietary protein was measured by the method described in Food and Agriculture Organization or World Health Organization in 2013, which is the ratio of available amino acids in the food compared with needs. The equation 5 was used to calculate the essentials amino acid score. The equation 5 is the one that is in the upper left side. And then the other one, which is in the right side, the essential amino acid index. Essential amino acid index, or EAAI, was calculated as described by Oser 1959 and Kobas in 2022 using this equation. And the next one, to predict the protein efficiency ratio, the author used this equation proposed by Oser 1959. Okay, the next one is statistical analysis. All statistical analysis in the study were carried out using R statistical software, version 4.0.2. Data were subjected to analysis of variants using completely randomized design. Significantly, different means were separated using Taki and a significance different, with R's agricole package. Pearson's correlation was carried out to determine the relationship among the amino acids and amino acids group at p under 0.05, 57 samples of each nutrient were correlated and presented in correlograms. The correlograms was plotted using the core-M test and core-plot function in core-plot R package. The correlation coefficients were presented in the percentage for space saving in the correlogram. Coefficient of correlations R were categorized as weak, R under 0.3, moderate, which is R equals to 0.3 until 0.49, and strong, which is R more or equals to 0.5. The data of each amino acid group were subjected to agglomerative hierarchical cluster analysis and presented with heat maps. R packages used for the computing and visualization of heat maps are Circlize, R Color Brewer, Dendextent, and Complex Heat Map. Prior to clustering, Euclidean distance method was used for dissimilarity measure, and data was standardized using the R-based scale function. Copanetic distance correlations were carried out to determine the most appropriate agglomerative or linkage method for each of nutrient group in their hierarchical trees. Consequently, average linkage method was used for the amino acids groups, except the total groups, which had centroid as the highest correlating method. The optimal number of cluster for each nutrient group was determined using NB-class function in NB-class R package. Next, we're going to be moving on the results and findings. So in the result and findings, the amino acid composition of the selected Nigerian cooked foods in milligram per 100 gram fresh weight or FW is presented in tables 2 and 3. I'm gonna show you the tables 2 and this is the table 3. Yeah, we're gonna review on it later. So yeah, indispensable, conditional indispensable, and dispensable amino acid were identified in the cooked foods. This is gonna be reviewing about the indispensable or essential amino acids. From the table 2, we get to find that this finding may be attributed to the ingredients used in the preparation of the cooked foods. All the soups and stews analyzed had fish, crayfish, and meat as part of their ingredients, while the composite meals were cooked with ingredients from pulses, cereals, and tuber foods group of all of which are good sources of these amino acids. However, the amount of these meals consumed per serving determines how much of the amino acids can be supplied to the body. In the study, leucine was the most abundant indispensable amino acids in the analyzed Nigerian meals except in Ila ala Sepo, Afang, Egusi, Kepekiri, Okbono, and Oha soups, where leucine was most predominant. Previous studies reported that leucine was the most abundant IAA in dairy, egg, fish, meat, processed foods, and their products in Portuguese diet, and some Cameroonian traditional dishes. Conversely, leucine was revealed as the most abundant IAA in fish species in Brazil. Indispensable amino acids are important in multiple metabolic pathways, playing diverse roles in humans' health, and must be supplied through the diet. Deficiency of IAAs may hinder protein synthesis and hence produce symptoms such as depression, anxiety, insomnia, fatigue, weakness, growth, among others. Habitual consumption of diets having low levels of IAAs may predispose consumers to low serum levels. Also, inadequate dietary intake of this indispensable amino acid had been associated with high risk of stunting, which is a major public health issue worldwide. Globally, stunting affects about 1/4 of under 5 children and 37% under 5 children in Nigeria, with 19% children severely stunted. Stunting has been associated with decreased survival, impaired cognitive and motor development, reduced economic productivity, and susceptibility to poverty in adulthood. This suggests that the analyzed Nigerian cooked soups and dishes might have positive impact on immune, mental, cardiovascular, and coronary health. Okay, so after talking the indispensable or essential amino acid, we're gonna be moving on and talk about dispensable or non-essential amino acids. Afang soup and chicken stew rank highest in glutamic and aspartic acid and contents, while afang soup rank highest in alanine and serine. Also, auha soup and groundnut soup rank equally with afang soup in glutamic acid contents. This shows that afang soup is not only predominant in indispensable amino acid, but also indispensable amino acid. Meals that were least predominant in DAAs might not be of health concern since this amino acid can be synthesized in the body. Glutamic acid is essential in protein synthesis, protein synthesis, conjugation of bile acids, and neurotransmitter synthesis. Glutamic acid was one of the most abundant dispensable amino acids in food proteins of plant and animal origin. And it serves as a source of energy for the brain, thereby improving brain clarity, mental alertness, and mood. It also plays a crucial role in chemical sensing in the gastrointestinal tract and other tissues. This suggests that the analyzed Nigerian cooked soup and dishes may have positive impact on growth, mental, and gastrointestinal. Is the amino acid groups in selected Nigerian cooked foods. Table 4 shows the amino acids group in the standardized Nigerian cooked foods in milligram per 100 gram sample of fresh weight. All the Nigerian cooked foods analyzed were significantly different at P under 0.01 for all the amino acid groups. Afang soup, followed by chicken stew, ranked the highest in TAA, IAA plus his aromatic, basic, and neutral amino acids. Similarly, afang soup and chicken stew ranked highest in branch chain and acidic amino acid, while chicken stew and groundnut soup rank equally with afang soup in sulfur containing amino acid content. This finding is contrary to the previous study where grains and grain products followed by meat and meat products were the major sources of amino acids in the current diet. Although the proportion of grains or meat consumed is a major determinant of the source of amino acids content derived from the diet. Also reported that branch chain amino acids rank higher in foods of animal proteins than foods of plant proteins. However, afang soup and chicken stew are soups which have meat and meat products as their major ingredients. This may explain why these soups are good sources of indispensable, aromatic, basic, neutral, branch chains, acidic, and sulfur-containing amino acids. On the other hand, miyan kuka and gebegiri soup rank the last in total amino acid. Boiled yam and garden egg sauce and avada rice and stew rank the least in branch chains, neutral, and sulfur-containing amino acids. Similarly, boiled yam and garden egg sauce rank the least in aromatic, acidic, and basic amino acids, while gebegiri soup and boiled rice and fish stew rank the least in total indispensable amino acid, with histidine. Avada rice and stew rank equally with boiled yam and garden egg sauce in aromatic and acid amino acids. Avada rice and stew is a cereal-based food prepared with unpolished rice grains, oil, has fish, offals, and co-skins animal proteins. Boiled yam and garden egg sauce is a tuber-based dish prepared with animal protein and garden egg, which is rich in dietary fiber as part of its ingredient. Also, miyan kuka and gebegiri soup are cooked with leafy vegetables and legumes, high in dietary fiber as their basic ingredients. All the soups and composites meal were prepared using animal proteins as part of their ingredients. Previous study conducted on the nutritional composition and heavy metal profile of Nigerian rice varieties revealed that avada rice had low levels of amino acids, especially lysine, arginine, methionine, histidine, threonine, cysteine, proline, tryptophan. Also, findings have shown that foods high in anti-nutrients tend to have lower levels of amino acids contents. Avada rice and stew has been reported to have high anti-nutrient contents. This may be responsible for the low level of amino acids observed in avada rice and stew and other foods having low levels of amino acids, particularly indispensable amino acids. Low level of IAAs and CIS in the diet could have negative impact on health. Okay, moving on to the next slide, it's the protein quality. Amino acid interrelationship is an essential protein quality indicator that reveals their potentials to predict chronic malnutrition and disease. All the Nigerian cooked food analyzed were significantly different at P under 0.01 for all the amino acid ratios evaluated except the BCAA or ARAA. In the study, beans and corn ranked highest in leucine to isoleucine ratio, while pangasup and mian kuka rank highest in leucine to valine ratio. However, akara and agidi ranked the least in leu or leucine, while chicken stew ranked the least at leu and valine ratio, but was similar to beef stew and ila ala sepo. Both leucine or isoleucine and leucine or tuvalin were greater than 1 in all the soups and dishes. The high proportion of leucine compared to isoleucine as well as leucine compared to valine in the Nigerian cooked foods may have beneficial effect in the management of chronic disease, including diabetes, and treatment of protein energy malnutrition. We can see it in table 5. This is table 5. Okay. Findings have shown that high leucine to valine and or leucine to isoleucine promote imbalanced branched chain amino dietary contents, reduced levels of valence and isoleucine in the plasma, which in turn predicts impaired brain uptake of multiple essential amino acids. Findings have shown that branched chain amino acids play important role in muscle protein synthesis and insulin resistance. Sulfur-containing amino acids are essential in the synthesis of intracellular antioxidants such as glutanoin and N-acetylcysteine, and they play a crucial role in the elimination of toxic metals in the human body. Low index of aromatic amino acids may increase the risk for incident hip fractures and aerial bone mineral density. Similarly, natural amino acids help to reduce the phenylalanine levels in the brain and increases neurotransmitter levels in phenylketonuria. The Nigerian cooked foods analyzed in this study had low proportions of sulfur-containing amino acids, while most of them had low proportion of aromatic amino acids. This implies that the habitual consumption of these foods without supplementation of sulfur-containing and aromatic amino acids may put consumer at risk of mineral toxicity, reduced intracellular antioxidant, increased risk of hip fractures incidents, and aerial bone mineral density. However, most of the analyzed cooked food had good proportion of branched chain amino acids. This may promote muscle protein synthesis and hence helps to mitigate against malnutrition. Okay, moving on to the next slide, it is the table 6 about amino acid scores of the selected Nigerian cooked foods. And the table 6 shows the amino acid scores of the selected Nigerian cooked food compared to the recommended values. The Food and Agriculture Organization, or World Health Organization, in 2013 recommended the use of amino acid score to evaluate protein quality. Amino acid score, which is based on amino acid requirement of different age group, is the reference protein amino acid pattern significant for calculating protein quality for dietary assessment. It is used to predict protein quality in terms of the potential capacity of the food protein to provide the appropriate pattern of dietary indispensable amino acids and also identify limiting amino acids. Indispensable amino acids not meeting the minimum requirement for human or having lowest quantity in the food protein relative to a reference protein is referred to as limiting amino acids. In this study, all the Nigerian cooked foods analyzed met the recommendation score for isoleucine, tryptophan, histidine, threonine, and aromatic amino acids except avada rice and stew, where threonine was below the recommended score. Findings have proven that complementing plant-based with animal-based food will provide a complete protein food, take care of the limiting amino acids, and improve protein quality in food. The consumption of this unstandardized Nigerian soup and dishes that met with the recommendation score for amino acids will supply good protein quality, which will be of beneficial health against protein deficiency disease. Habitual consumption of this cooked food having limiting amino acids may predispose consumers to deficiency disorders associated with those amino acids. Therefore, amino acid supplementation is crucial to prevent deficiency disorder of amino acids limiting in the evaluated Nigerian cooked foods. Okay, the next slide, we're gonna be talking about the clustering of the amino acids content and their groups in the selected Nigerian cooked food. As you can see in the figure that I present in the presentation, it's about the cluster for the indispensable, dispensable, conditionally indispensable, and groups D of the nutrients or amino acids in the cooked food. So meat and seafoods have been identified as good sources of indispensable amino acids. However, the combination of meat and seafood in the diet results in higher concentration of essential amino acids profile. This suggests that combination of meat and seafoods, including combining different types of seafood, may have higher concentrations of essential amino acids compared to meals having one animal protein. So if we have multiple animal protein sources in one food, that is gonna be better or greater to provide these essential amino acids rather than only one animal protein sources. Also, afang soup is similar to oha soup in indispensable amino acids, suggesting that these soup are not just higher in indispensable amino acids, but also indispensable amino acids compared to other soups and composite meals. However, avada rice and stew, which was a sole member of cluster 1 for conditionally indispensable amino acids, had offals and co-skins in addition to fish as protein sources, while afang soup, a sole member of cluster 2 of CIA, had beef, fish, crayfish, and periwinkle as protein sources. These animal protein sources are not only rich in indispensable amino acids, but also good sources of proline, glycine, and arginine, which are conditionally essential amino acids. Habitual consumption of these soups during psychological conditions such as pregnancy, adolescent growth, preterm burns may be of health benefit. Foods having similar amino acids composition can serve as varieties for consumers undergoing diet prescription associated with amino acids. This information will be beneficial to nutritionists and dietitians in meal planning and diet prescription aim at managing health and disease conditions that are associated with protein quality. Consumption of foods with better amino acids profile habitually will promote protein synthesis and hence reduce the burden of protein malnutrition among consumers. Okay, moving on to the next slide, we finally reached the conclusion of this study. So this study provides information on the amino acids composition and protein quality of Nigerian foods. The major source of indispensable, conditionally dispensable amino acids is afang soup, which is a vegetable-based soup prepared with varieties of animals' proteins. Composite meals having low quantities of animal proteins had low level of the indispensable amino acids. Also, the Nigerian cooked foods analyzed in the study contain more dispensable than indispensable. And they contain higher acidity compared to basic amino acids. In addition, habitual consumption of the cooked food may have beneficial effects against diabetes and protein energy malnutrition due to its high proportion of leucine compared to isoleucine and valine respectively. Most Nigerian foods having high branched chain amino acids content might mitigate against impaired growth and muscle wasting when habitually consumed. Most Nigerian cooked meals have low predicted biological quality, although most often had good amino acid score and high predicted protein efficiency ratio. Furthermore, afang soup had the higher quality of indispensable amino acids compared to the other group and composite meals. Frequent amino acid co-occurrence in most Nigerian meals may be useful to predict amino acids for which there are no nutrient data in Nigerian foods. Clustering of Nigerian meals are useful as information essential for meal planning in the management of disease conditions as well as food composition database. These findings are essential for dietary assessment and formulation aimed at reducing the burden of malnutrition and associated disease among Nigerian population. Besides, habitual consumption of most Nigerian meals without consuming other sources of sulfur-containing amino acids and aromatic amino acids may put consumer at risk of mineral toxicity, reduced intracellular antioxidants, hip fracture incidence, and areal bone mineral density. So yeah, all of the 19 selected commonly consumed foods in Nigeria had a great rate of amino acid, but most of them is low. They mostly low in the sulfur containing amino acids and aromatic amino acids. So they have to habitually consume the meals that also contain sulfur amino acid, also aromatic amino acid, to help them to reduce the mineral toxicity, reduce intracellular antioxidants, and hip fracture incident, and areal bone mineral density. So this is all my journal presentation. I know this is my very first journal presentation, and hopefully this is gonna be leading to a better presentation in the next future. So thank you so much, and see you in the next journal presentation. Bye.